So now we are moving to the next part of the day, which will be the next several sessions or few focused on spinal cord stimulation, neuromodulation strategies. And just to, again, if you haven't read through the Navigating the Symposium, our thinking and idea was we wanted to hear from a few scientists thinking about what's happening in a lab. What are scientists slash clinicians thinking about around spinal cord stimulation? You all have heard about it. We wanted to dedicate some time to it. Obviously, it's in the top of a lot of people's minds, and primarily focused in these sessions on transcutaneous and epidural stimulation. And we'll explain what that is. These presenters will. Thank you. And from there, we're going to move into a little more clinical focus with a few therapists working very practically, I think, and strategically. How do we translate what we've heard about spinal cord stimulation into the clinical setting, clinical focus? What does this look like? It gets a little bit to what Kristen's question was in some sense, I think. And then from there, we'll go back and then we'll hear from a number of folks with spinal cord injuries who have experience with spinal cord stimulation, representing both companies that are emerging that you've heard about, like Onward, Spinex, and Anuvo, and also clinical, sort of pushing the envelope clinically. And then we'll finish out the day kind of going back to some strategy questions around that. So leading up to that, our first presenter is Peter Grahn from the Mayo Clinic. Peter, come on up. Peter's here in, yes. I could do it if you want. Okay, or you want one of your, are you friends enough with them? You don't want, no, okay. All right, yeah, you can count on me, man. Yeah, I'll turn it on. Wow. That was rough, man.
All right. Testing. Can everyone hear me all right? All right, so like 6 months ago or so, Matthew asked me to give this talk on science, and I've gone back and forth a couple times on whether to stick strictly to science or to try to, there's really no way to parse it out from my own journey through spinal. If it's not obvious, I have a spinal cord injury, if you can't tell. So to separate them out is kind of difficult. So this is a hybrid presentation that kind of wraps it all together, my own journey, and then the science that our team is doing at Mayo. So you can advance one more slide, Matthew. And so to start, I just wanted to thank everyone, kind of do a presentation backwards. We usually thank them at the end. I wanted to thank some people at the beginning. United by Paralysis for the opportunity to come here and share my story and our research today, as well as that good-looking guy next to the U2FP symbol, Matthew, for his friendship over the years. And you skipping? We're done? For his friendship over the years and his ambition, his drive to advocate for SCI research and translation, as well as his willingness to ask the difficult questions that others in the room maybe aren't willing to ask. Matthew's always right there, bugging us and pushing us to try to think differently, I would say. And then I got to give a shout out to my family from my own journey perspective of supporting me through my own injury and getting me to where I am to here today. My parents are actually in the crowd with my sister. My dad took time. My dad, yeah, over there. My dad took time out of the cornfields to come today, so thank you. And then our rehab team, our research team at Mayo, the collaborators we have in pain medicine that help with doing our current study, which involves percutaneous trial leads, as well as the rehab team, the engineers. Many of them are here today. If you cross paths with somebody with a Mayo tag, they're more than likely, we're working with them in some form on the spinal stimulation projects. Our funders, the NIH, our current clinical trial is funded through an R01 that was awarded in 2019. So that's really what kickstarted the project we're working on now, as well as we have a few projects of modeling that are supported by the Office of Higher Education, the Spinal Cord Injury, Traumatic Brain Injury Program. The Jack Jablonski Foundation has funded us for several years, especially with our pilot project that involved 2 people, and then a subsequent project that was ended prematurely by COVID. Ended a lot of things, but it was unfortunately ended one of our projects that was going strong. And then that picture of the guy with the gray hair, if you don't know him, Reggie Edgerton, he's kind of like the Yoda of epidural stimulation, I like to think of, in a good way. He collaborated with us and when we initiated these projects at Mayo, and really brought his team on board that was filled with experts that had previous experience with epidural stimulation, and that really blossomed into a collaboration with Dr. Dmitry Sayenko, who left Reggie's lab and started his own lab. There's a picture of him there with his team at Houston Methodist, and so he's a collaborator on our project, and we collaborate with him on other projects, and his insight into what we're working on is very valuable. And then from the industry side of things, I'd like to thank Abbott for their partnership with us. They've donated research devices, supported us with our programming, and really been an excellent partner. Next slide. So just an overview of what I'm talking about. So I wanted to summarize the last roughly 6,648 days of living with spinal cord injury. The prior work that we initiated these projects at Mayo in the clinical setting, and then what that has expanded into with our current projects. That's the main theme of this presentation. Next. So for my own personal story, in 2005, August 7th, I was swimming in Green Lake, which is about 2 hours west of here, and dove into a sandy region, very shallow water, broke my neck, C5. From there, I was airlifted to St. Cloud Hospital, had spine surgery, fusion of 456, was determined that I was Asia B, so I have fairly decent sensation, proprioceptive. Ball bladder sensation is all intact still. And then I spent 10 months in rehab. So I went from St. Cloud to Abbott Northwestern, Sister Kenny, it was called at the time. I know they've changed names down there a lot between Sister Kenny and Courage Center at the time. So it was 10 months there. And then I graduated from rehab. I was actually thinking about an undergrad or when I was starting undergrad, going into PT school. And then instead I just did PT for a year. So thought that was enough of that. But if anybody's experienced the halo, like being in prison, feels like jail cell bars. I had that for 3 months. I think I got it off like the day before my birthday. So that was a great gift. Next slide. And a fun fact about me is Minnesotan. Yeah, you betcha. I lived the first 18 years of my life in Wilmer, Minnesota. Had my injury, lived in the cities for a year, then moved to Southwest State, Marshall, Minnesota. I was there, met my wife in 2006, 2011, then transitioned to, when I graduated, I transitioned to Rochester Mayo Clinic, and I was there from 2011 until currently 2016. I earned my PhD in neurobiology of disease. And we've been working on these clinical trials about the same time that I graduated is when we started the work that I'm going to present here today. Next slide. And so our prior spinal stimulation projects, you can go to the next one, Matthew. So in around 2015, 16, when we were collaborating with Reggie, we decided through some internal funding as well as some other funding partners, we decided to pilot study 2 participants with a permanent implant made by Medtronic to treat pain. And the idea was we would put them through 6 months of rehab alone before they ever received any type of stimulation, just to make sure their injury was stable. It was somewhat of a replication study of the work that came out of Louisville over the last decade. We just wanted to get it started at Mayo, try to see what we could learn. Our participants showed no real signs of improvement during those 6 months of locomotor training. So we moved forward with implantation of the 16 electrode array, the Specify 565, and then that was connected to IPG in the abdominal area. They went through about a year of rehab with stimulation, both of these participants, and so this is the example CT and MRI of our first participant showing his injury site on the top, and then the array at the bottom just below his spinal hardware from his injury. And the reason that we place the array there is to target the muscle groups of the lower extremities or the lumbar enlargement, L2 through S1 area. For stimulating different standing, stepping activities was the goal. You can go to the next one, Matthew. And once we implanted them and we began stimulation, we really put them through a whole dynamic array, a vast variety of rehabilitation paradigms where we had them supine, sideline with their leg elevated, trying to do step-like motions with no gravity effects, in a treadmill with body weight supported, and overground, trying to work on trunk balance. It was like every day it was something new, just trying to challenge the system and see what we could improve. Next. How do we play this? Or Matt, maybe you want to come up and just like do an interpretive dance. Okay. So this video here is showing after the majority of the rehab, almost a year. Here we have stimulation off. You can see that they're having the rehab team on each side is having to step his legs through. He's got body weight support on. The channels that are coming through here, blue is flexors of your left leg on the top 5 or 6 channels, and then blue flexors on the left leg. Sorry, I had that backwards, left and right. Anyways, but what it was showing was that there was very little activity. Most of it was reflexive from just moving his leg. Here with stimulation, you can see he doesn't have body weight support on. There's no real assistance from the therapist. He does have his arms on parallel bars. So he's holding himself up and he's doing weight shifts, manipulating his trunk with his arms. But they're independent steps. It's not walking, they're independent steps. And the treadmill's moving slower than what would be considered like a productive ambulation speed. But the EMG patterns are not normal, but they are bursting in a coordinated fashion with the stimulation pulses that we're delivering. Next. And so what we took out of that was that if we stimulate on one side of the array, just with a few of the contacts, we can activate specific muscles of that leg. If you've heard of spatial temporal stimulation, where you're targeting different motor pools, here we're more generally activating one leg with a program, and then immediately after, like within milliseconds, we're activating the other leg through sensorimotor circuitry of the spinal cord. So if we alternate those stimulation paradigms as shown here, you can activate the gastroc and soleus, let's say on the left, through a monosynaptic reflex response, while the other leg is still silent or potentially coordinated to, if you're trying to cause flexion of one leg, the other leg is causing extension. And so this is just preliminary data that we gathered from our first 2 participants. Next slide. And this is emphasizing that fact again, when we get them upright and over ground or on a treadmill, and we apply this interleave paradigm of stimulation, if you look in the bottom right there, it's showing alternating pulses of stimulation, and the muscle groups from each leg are responding to the corresponding pulse on that side. So it's causing like a coordinated alternating effect that they either learn to manipulate or their descending drive learned to coordinate with in the spinal cord to cause this stepping movement. Next slide. And so that was kind of the take-home discovery that we came away from our first pilot study. And so the question became like, how do we expand on this work? What do we want to go after to advance the knowledge of spinal stimulation? And the first step in that whole process is grants. So writing grants. But from what we learned from our first study is that the fully implanted system is expensive, which then budgetary wise, when you're writing a grant, it limits the number of people you can study to a number that may not even be valuable when you come away with your statistical analysis of the data. And then personally, as I was developing the grant, I had ethical concerns or considerations as far as like, I'm a new investigator early in my career. Do I really want to put permanent devices in people? And then after the study, wonder the rest of my life, their life, like, what are they doing with these? Are they safe? Does it even work anymore? Like, was this a waste of everyone's time? Don't know the long-term effects. So I turned to the idea of placing temporary leads or percutaneous leads that are developed. But so if you're familiar with the therapy for pain that these devices were developed for, before somebody gets a permanent implant with the IPG and the array, insurance companies and clinicians will place temporary leads. So they're placed with just a needle. The back end of the electrode comes out the back, and it's packaged under adhesive bandaging. And then they go through approximately 2 to 3 weeks of stimulation to see if it treats their pain. So I wanted to leverage these temporary leads, but place them into people with spinal cord injury and look at the short-term effects of maybe 10 days of rehabilitation, as well as at the end of it, we explant them, they slide back out, and the person goes on their way. They don't have a permanent implant that the study involvement shouldn't influence their participation in future studies. And I think, Matthew, I think you said it was a bad idea, right? Nobody would sign up for it because they all wanted permanent implants. But I mean, that's fair enough. But we went through it. We went forward with it anyways. I didn't listen to them. Next. So just kind of covering our current research, we're doing a clinical trial of that study where we're placing the percutaneous leads. It's funded by NIH. And then we're also doing a computational modeling study that's funded by the Minnesota Office of Higher Education, so one of the main questions that we're coming away from every day when we leave the lab is like, how can we improve the stimulation? How can we better understand or develop a tool that guides where we stimulate and the timing of it and what the most appropriate parameters are? So we got a grant to try to visualize or analyze all of the parameters that we put into the spinal cord for stimulation, as well as the readouts that we're getting and the imaging that we're collecting, and trying to better understand what happens when we stimulate, as well as how can we target better to different areas of the cord. Next. So yeah, we put in this grant, and then COVID hit as soon as it got funded. So we spent like a year and a half wondering, is it safe to get into lab? Should we bring people into lab, like, and risk them getting COVID? And there was so many unknowns that like it was just impossible to get started with the grant. But now we're running at full steam, luckily. But we lost a significant amount of time during COVID from that. Next. And the overarching goal, as I said earlier, is to study the sensorimotor activity that's produced by spinal cord stimulation in a large number of humans with a variety of severity of SCI. The first aim is more of a mechanistic electrophysiological aim, where we're stimulating the epidural surface, as well as we place electrodes along the dorsal roots, and we're stimulating there and looking at the interactions that occur when you send pulses to each area. And the second aim is the more exciting aim from a functional standpoint, in looking at temporary stimulation with 10 sessions of rehab. Are there any changes that occur in somebody's function, as well as during those 10 sessions, that brief period of rehab, can we identify stimulation parameters that are good enough to achieve some level of function? Because at the time, we didn't know how long it would take to even familiarize ourselves or the participant with epidural stimulation through these percutaneous leads. So step one, after we got the award, we got FDA approval of an IDE to use the Abbott device connected to a clinical research stimulator and recording device made by Ripple. And the overall goal, or the total number of subjects we want to study, is 32 from all severities of injury, as long as their injury is above the area that we implant the electrodes. So T11, T12 region is the target area for the electrodes. Next. And this is just kind of a schematic of what a cartoon of where we implant the electrodes. So you can see we place 2 electrodes in parallel to midline from like the T12, T11 actually to L1 region of the cord, or L1 region of the spinal vertebrae, which spans that L2 to S1 segments like the previous slides had shown, targeting the lower extremity muscles. Then we move down and we place 2 electrodes on the dorsal roots at L4 or L5 region. The idea being those roots are innervated to the lower extremity muscles, so we can look at their activity and sensation as we stimulate. Next slide. And then we put them through 10 days of rehab, and this is just a setup of what our stimulation and recording device looks like. So we have 32 we have channels that run to the epidural electrodes as well as the dorsal electrodes, and then it records back synchronized data. Next. And I think I already talked through this. So we're looking at activation between the epidural and dorsal root electrodes and how they interact with each other. I just want to get to the fun stuff. So you can go to the next one. Here, we can show these videos. Can we play these videos? Please, one at a time. So this was a participant of ours in the study with cervical level motor complete injury, and we're using epidural stimulation here. And he's stepping on with minimal assistance. Normally he would need full assistance at the legs, and you can see the rehab team barely has their legs on his ankles as well as near his knees. They're not really helping with swing phase, just kind of supporting during stance phase. And I think this was maybe the 5th day of rehab with stimulation, somewhere around there. And then in this other video on the right side, so this participant, this is probably like the 5th day as well, is stepping. He's got body weight support that's holding him over ground of his upper body. He is also motor complete, so without stimulation, he wouldn't be able to take any steps. You can see here, Megan's just kind of helping with guiding his leg through. Stance phase is pretty independent, and then just helping with his trunk stability. And so I should wrap it up. Okay, sorry. But I just want to comment that like the outcomes that we're achieving are variable across people depending on like their DEXA score, if it's even safe to get them up, dependent on their injury level, if they have strong enough arms to support themselves. Next is just a basic overview of our computational modeling work, which you can see a poster tomorrow. Anders will be presenting at the poster session. So he's the expert to ask, not me. I think that's it. How'd I do?
Mistake. As I mentioned, having Peter here is an example of, I'm going to pull this forward. Just got out of the way to make room for. You know, very, very simply put, you know, Peter just shared an example of, okay, we're trying to figure out how to use this thing, how to make it work, and test it in a variety of ways. And then there's the other part of trying to figure out, okay, well, with the things that work, how does this make sense in a clinical setting, right? That's what you all are here for. So welcome Jill Wecht, Noam Harel, Jacob Goldsmith from New York. Oh, here.
Oh, hello. Okay, this.
Yeah. And that's the green arrow.
Yeah. Good. All right. Hello. I'm Jill Wecht. I'm a 58-year-old female with, I think, salt and pepper hair. I have a black sweater on, and I've been studying the consequences of spinal cord injury for 20 years. But I'm not going to talk to you about that today. I'm going to talk to you with my colleagues about one center's trajectory through the past with clinical academic research into the present with our young career development awardees, and then into the future on how we think, some of the hurdles that we're going to encounter and how we think we as a center would like to change the paradigm of purely investigator-initiated research into clinical translation that improves clinical care, health, and lives of people with spinal cord injury. Okay, so So this is our past. We had an exciting start in the 1990s with Dr. Bauman, William Bauman, and Dr. Ann Spungen, who initiated the center. And we went we had some exciting, promising, initiated research that was translated in, well, from two to a multi-center lab that led to a lot of independent, multi-dimensional research programs with various focuses. And we're a combination of the medical consequences of spinal cord injury. So we studied all the secondary consequences, not necessarily the skeletal muscle paralysis and the inability to walk, but other things like bowel and bladder function, like autonomic dysfunction, and like endocrine dysfunctions. Sorry. Okay. And that work has translated pretty nicely into some clinical practice implementation. So numerous investigators within the center have contributed to clinical practice guidelines like the endocrine guidelines and like the autonomic guidelines, as well as the international data sets for how we're going to measure and record progress or even the degree of injury over time in people with spinal cord injury. And we've also at our local center implemented routine DEXA scans for bone mineral density, as well as oral glucose tolerance testing for dyslipidemia. And we've also had plenty of academic success. So this is just a snippet of our grant funding. As you've heard this morning, we're in a constant treadmill of trying to have more funding to keep the lights on. Publish or perish. We're doing a lot of work. We're publishing, constantly. We're always on that grind of not only getting the money to do the work, but also dissemination to the academic environment, right? So how well does that actually translate into, are people seeing the progress of what we're doing? Are they using the interventions that we're studying? Are we getting it into clinical practice to help the lives of people with spinal cord injury? But we spend a lot of time on bureaucratic nonsense, on reporting, and it's all important. I'm not saying it's like without merit, but it does take up a lot of our time. And what it leads to is cycles of funding, reporting, publishing, funding, reporting, publishing, takes away from, again, that dissemination, that ability to translate into the clinical practice and improving the lives of people with spinal cord injury. So this leads to silos. And this, we believe, is one of the major problems with the current academic setting, is that we're in our individually focused areas and we're really productive. We're doing great work, but we're not able to see beyond the barriers of our silos and collaborate. We're not doing as much collaborative work that's going to really work toward improving, again, the lives and the clinical care of individuals with spinal cord injury. So this is our past. We've been in silos. We've been very productive academically. We've had some success with clinical implementation, but too much time has been spent on those pursuits, on the metrics associated with academic success. So that leads to and we've had inconsistent community involvement. We need to do a better job of hearing from our community and implementing their needs, prioritizing their desires in our research. And so that is, again, with our research goals, we want to institute those practices in the clinical setting. We have we want to focus more on personalization of rehabilitation, but we do have some real barriers to overcome. And these are unfortunately not able to be covered by grant dollars. We have barriers, including like transportation, lodging of our patients. We want to follow them long-term. Well, how do we get them back? How do we keep them engaged? How do we keep them healthy so that they can continue to be engaged and continue to be compliant with our research and in their lives? But that's not going to be funded. That's not going to be covered by grant funding. So this, we believe, is time for a change. So part of the change that we're instituting is young investigators. We're trying to bring more young enthusiasts. Oh, if you can't be introspective, then you need to look at yourself. Okay, a little meme. But we're trying to bring on new, exciting, young minds so that we take our old dodgy minds and we mix up with some new and exciting, innovative people. And so one of them is here on stage with us. Jacob Goldsmith joined our lab a year ago, and now he'll talk to you about his journey and a little bit of his science.
Morning, everyone. Name's Jacob Goldsmith. I'm a 6'1" white male with a blue suit and green eyes. So thanks for the introduction, Jill. I am a postdoc working with Noam and Jill up at the Bronx. And my background is in cardiovascular and exercise physiology. And I got my PhD from Florida State University in 2020. After that, I did my first postdoc in clinical spinal cord injury research. And this is at a different VA medical center. And there, I got to know some really cool participants. Shout out to Oh, sorry. Not holding it close enough. PhD, Florida State 2020. Then I did a 2-year postdoc at a different VA center, looked at various interventions to improve health and function after injury. There, I got to work with some really cool participants. Shout out to Josh, who's a really good wheelchair rugby player. He played on the USA low point team and also the all-veteran Oscar Mike team. And so through him, I started getting involved in some volunteering at Oscar Mike and then also the PVA games. And during this time, I was also lucky enough to have a fantastic lab mate, Dr. Tommy Suter. And his passion for spinal cord injury research was infectious. And he's actually the person that introduced me to Matt and U2FP. And so through my journey, it was meeting people like these amazing participants and people like Tommy and Matt that kind of solidified for me that doing this type of research to improve health, function, and quality of life after spinal cord injury is truly the right path for me. But as Jill mentioned, as academic researchers, we have certain metrics that we have to hit. So essentially, we have to be publishing papers and we have to be writing grants. And I've seen a side of research where papers are written and grants are written primarily for these metrics and to get good publicity. And so I found myself questioning in this process, what is the actual impact of this work that I'm doing? And during this time that I was struggling, I actually met Matt at the Asia conference in 2022 and had some really good conversations about good researchers in the field that are doing good work. And he actually recommended Dr. Noam Harel. And so long story short, I went to my first U2FP last October. And the next month in November 2022, I started my work at the Bronx with Noam and Jill. So overall, my interest is in using testosterone replacement therapy, neuromodulation techniques, and task-specific exercise to try to improve both motor control and cardiovascular function after injury. And I recently submitted my first grant, and it was selected for funding dealing with testosterone replacement therapy. So our center has actually done some work along with other labs to show just how prevalent low testosterone or hypogonadism is following injury, with up to 45 to 60% of males exhibiting low testosterone. Importantly, this can exacerbate many of the deleterious effects of the injury itself. And so this figure is the percentage of low testosterone observations. In the gray bar is spinal cord injury, and in the black bars are the uninjured controls. And as you can see across the age span for both total testosterone and the amount that's free or unbound, low testosterone is much, much more prevalent following injury. Our center and other labs have also done work to show many of the consequences of having low testosterone, and also that replacing testosterone or improving those testosterone concentrations to a normal level can have several benefits, including improving energy expenditure and body composition. But there also seems to be some really powerful neural effects that are relatively less explored. And so my project is kind of pulling together different aspects of our research. So neurorehabilitation, cardiovascular autonomic function, and endocrine function, essentially breaking down some of those silos that Joe previously mentioned. And so my project is essentially 2 parts. The first part is a larger cross-sectional study looking at the relationships between testosterone profiles and neurophysiological and cardiovascular autonomic function. And then the second part is a smaller experiment, actually using an intranasal form of testosterone replacement therapy and examining its acute effects on neurophysiological and cardiovascular autonomic function. So overall, there are numerous benefits to increasing testosterone concentrations to a normal normogonadal level. And this may include promoting neural health to improve both motor control and cardiovascular function to improve rehabilitation following spinal cord injury. Thank you.
Everyone. Hi, everyone. So I'm Noam Harel, and I think I basically look like Jake will look in about 20 years after he shaves his beard. Similar height, both white. So you just heard from part of our center's future, and we've heard a lot of great talks already today. We are trying to be more introspective and self-critical. The grades out there for actual clinical achievements are ranging from C, D, or lower. So we're trying to figure out what are the things that the good things that we've done as a center that we can retain and try to keep our eyes focused on the bigger prize as we move toward the future. Okay. So again, I think we all we all have the same goals or the same overall metric in mind. All the stakeholders in the guitar string, we all want to help, you know, for quality of life and for function after spinal cord injury. But that's the forest, and we tend to lose our sights in the trees of all these smaller metrics. So we want to try to overcome some of these distractions, and that's going to take change in culture. So we happen to be a clinical research center. We're based inside a hospital, and yet when people come into the spinal cord injury clinic, they tend to not hear as much about the opportunities they may have for doing research. And that's on us. The clinicians are not, we need to help clinicians get more in tune with our research. So we need to improve the amount of protected time that clinicians have to be able to get immersed in research themselves. And we're starting to set up exchanges hosted by different parties in alternating fashion, where we can both become better at understanding the other side's world and make one world out of that scenario. So obviously this comes up a lot. Outreach. I'm very curious what people here with lived experience think about this. Do you really feel like this is getting you involved as part of the team or not? Because sometimes I worry that we're not actually living up to it. So what I think would help is actually having people with lived experience not just be advisors, but be part of the team. You know, nothing replaces working with someone every day, no matter how many Zoom meetings you have. So we hope that ourselves and other places will have more people with the right experience, lived experience on the team. And yes, it does require focus groups, which does mean Zoom sessions. We want to have standing focus groups with multiple different kinds of stakeholders, people with lived experience, but also clinicians who are not researchers. Because not only will this help us set our priorities straight, but it'll help us hopefully do things in a way that actually gets across that failing gap that Melissa Miller mentioned of translating this in a realistic or feasible way to the clinic. I'm going to try to speed things up here. Okay. We, as Jill mentioned, we need general funds for this. Scientific grants fund specific scientific questions. These are general issues. Okay, another general issue that's a problem for people with spinal cord injury, and it's a problem for our research. Transportation, whether it's public transportation in New York or any mode of transportation anywhere, it's very difficult for people to get to our center or elsewhere, and this impedes our research progress. And that's only if you live locally. If you live far from a research center, you may not have any way to pay for the lodging that it requires. So we're looking for general funds for that. And I do want to emphasize this: general health, UTIs, pressure sores, autonomic dysreflexia. These are things that hamper people's lives after spinal cord injury. They happen to hamper your participation in research. So we think improving overall health is critical for any type of neurorehabilitation, autonomic rehabilitation, or intervention. So we're inspired partly by a local gym near us in New York called Rehabilitation. We are striving to build a clinical rehabilitation facility to help keep people's general health as optimized as possible. And then on top of that foundation, we will add, you know, the best research interventions we can. And that will improve people's eligibility for our studies. It will improve people's interest in our studies, and it hopefully will improve their response to our studies for them to be in the optimal health. But it takes money. Okay. So again, you just heard from Jake. Mentoring is something universal in any workplace, but we want to mentor people not just to become their own investigators. We want to mentor a certain culture. So we're working on that. No better example than that movie. Okay, and that will maximize research value for everyone. So again, just to summarize, these are things that we think will hopefully help our own center improve and get to the real metrics, which is actually improving people's lives. So we need better stakeholder input, including clinicians who are not researchers. We need general funds to support things like transportation and lodging. We are 100% enthusiastic about sharing data in repositories. Adam Ferguson is one of our heroes. He has spoken here before. And again, we're trying to work on developing a culture, and really an ultimate goal is to become an actual clinical rehab facility that has world-class research on top of that. And again, that's the way to build a more sustainable clinical research partnership that really keeps the eyes on the grander prize. So just to wrap up, we're at a VA research center, but you do not need to be a veteran to participate in our research. And obviously we couldn't do anything without all of our great coworkers and collaborators and our funders. So thank you.
Thanks, Joe. Well, you heard, I think Noam said it, I don't know how many times. He wants input from you, so he'll be here for the next couple of days. Go find him, talk to him, and Jill and Jacob. Thanks, you three. Next, we're gonna hear from Ryan Solinsky at the Mayo Clinic. And one thing I wanted to mention, again, if you haven't read, Ryan has a recent publication asking some questions about, particularly related to autonomic dysfunction and spinal cord stimulation, which I think is somewhat provocative in the current field. So welcome, Ryan.
This mic turned on. There we go. Excellent. Giant green button. Make it go back. All right. Wonderful. As Matthew mentioned, my name is Ryan Solinsky. I'm a spinal cord injury medicine physician scientist down at Mayo. So I'm coming at this a little bit differently. I'm coming at it more from a clinical standpoint. I see patients in outpatient clinic with spinal cord injury, take care of the inpatient side, and then also do some research. I'm really excited for the opportunity to discuss autonomic dysfunction after spinal cord injury and different ways of characterizing the deficits, the effects of spinal cord stimulation. I would say one of the points that was made earlier by Naomi Kleitman about the field shifting from being very motor-centric and looking at motor outcomes to all these other problems that occur after spinal cord injury in the last 10 years or so has been really pointed and noticed. When I give this talk, outside of this sort of context, I always start it like this. When people think about spinal cord injury, they classically think about those outward manifestations of paralysis. And while that's true, everyone here knows that that is far from the entire story. In fact, when I'm giving this to a less educated audience, they're often surprised when I tell them that in a typical hour-long outpatient clinic visit, we spend maybe 5 minutes talking about motor and sensory deficits. Instead, we're spending a lot of time talking about those secondary complications that Dr. Harel brought up. Things like neurogenic bladder, right? Inability to empty your bladder, problems with urinary tract infections, problem with incontinence. We talk about neurogenic bowel, inability to empty the bowel without taking a lot of medications, to the point that in clinic, if someone says that they have a bowel movement in less than an hour, we consider that a win, right? Outside of that context, we would never consider that a win. We talk about problems with the skin, problems with pressure injuries that people develop. We talk about blood pressure problems, either blood pressure that goes too low with orthostatic hypotension or too high with autonomic dysreflexia. These are all major concerns for people with spinal cord injuries. No one wants to spend 3 months in bed rest healing a pressure injury or be in and out of the hospital with a urinary tract infection. The other important thing about these secondary complications is all of their physiology is rooted in the autonomic nervous system. As I started out, we focus a lot in research on motor and sensory outcomes, getting people to move again, which I think is 100% needed and appropriate. But sometimes we're lacking in understanding these other aspects. The autonomic nervous system is a little bit different than the motor system. In the motor system, it's all about sending this descending control down from your brain, telling those muscles to move and turn on. The autonomic nervous system is much more about balance. You've got the sympathetic nervous system, or the sympathetic component, which is very much a fight or flight response. And you've got the parasympathetic component, which is kind of rest and digest. And depending on the context that you're in, you might need more fight or flight, run away from a tiger, or you might need more rest and digest and sit and watch football. Maybe that's more of this, depending on if you're watching, which team you're watching. I think the illustration that shows this balance or imbalance best is probably autonomic dysreflexia. It says autonomic in the word, so it must be related to this. The way I like to explain autonomic dysreflexia is like this. Before a spinal cord injury, you have an intact neuroaxis. So your brain connects down to your spinal cord, connects out to your peripheral nerves, out to your organs. So if you have some mildly painful signal, let's say there's some fullness in your bladder, that signal starts out in the organ, goes out through the nerves, and gets to the spinal cord. And when it does that, it does two things. It gets there and says, hey, there's something mildly painful in this area. I don't want to wait for that signal to get to the brain. Let's reflexively tell all the blood vessels in the area that they need to squeeze. Maybe we're bleeding to death. Maybe this is something really bad. It then sends that signal up to the brain, and the brain says, nope, false alarm. Your bladder's just full. You shouldn't have had 3 cups of coffee. I know it's like unlimited coffee, but like we've talked about this. It sends a signal back down. I don't know if it's actually unlimited coffee. I should make that caveat. It sends a signal back down the spinal cord, out to the peripheral nerves, to the vessels saying, hey, false alarm, you don't need to squeeze. In this case, blood pressure does not appreciably increase. However, after a spinal cord injury, you can lack continuity in that neuroaxis. So while the first part of that reflex arc is still intact, saying, hey, maybe we're bleeding to death, squeeze all those blood vessels closed, that signal never makes it up to the brain. And correspondingly, the signal from the brain never makes it down to those blood vessels saying, false alarm, don't worry about this. This means that blood pressure can get higher and higher and higher until it reaches dangerous levels. There's been cases of autonomic dysreflexia where people's systolic blood pressure, that top number, has reached 325 millimeters of mercury, which is just an absolutely incredible number, especially with that sound effect. That was not planned, but I assure you, it is that scary. And people understandably have significant consequences to blood pressure that's that high, right? There's been cases of stroke, people bleed into their eyes, people have died from autonomic dysreflexia and that high of blood pressure. The other reason I wanted to bring up dysreflexia, as I mentioned, the autonomic nervous system is all about balance. And in autonomic dysreflexia, when someone has an autonomically complete injury, there's imbalance. Below the level of injury, it is fight or flight. It is all sympathetic, squeezing those blood vessels. And above, the body doesn't really know why this is happening. So it's all parasympathetic, rest and digest, try to pull you back into balance. How do we assess autonomic function in clinic? How does your physician, when you see you, try to get a handle on what your autonomic function is? I would say as a clinician, most of us do this. We use the pin, we use the cotton swab that everyone's familiar with. We do an INSCI exam, an Asia exam, and say, all right, where is your sensory level? Is it at T5, right? We're using just the pin and the cotton swab at that point. We're not even looking at motor function. All right, it's at T5. We have the cutoff of at or above T6. You're at risk for autonomic dysreflexia. Oh, nope, it's a little bit lower. It's at T7. It's below that threshold. I guess maybe you don't have problems with your autonomic function. You can see where this starts to become a problem. We're looking at sensory assessments to try to look at a whole nother part of the nervous system, the autonomic nervous system. So we've tried to come up with a way of addressing this. And the way we've done it is coming up with some objective tests of actually looking at the autonomic nervous system, challenging it, and looking at the responses, basically determining how autonomically complete or incomplete a spinal cord injury is. To do this, we need to use a different set of tools than necessarily just testing sensation and strength. Sorry, my slides are over. Backing up one second, how does that motor sensory assessment do? It's not great. If we look at level of injury, higher levels of injury have more autonomically mediated secondary complications, problems like urinary tract infections. It only predicts about 12% of the variability. So it really doesn't correlate well in terms of motor sensory completeness, so the Asia impairment scale, that also does not do very well. So we really need new tools to examine the autonomic nervous system in a systematic and objective way. And that gets to the point that I was just trying. So to do that, we need to look a little bit differently. We need to look at characterizing the ability of an individual to activate their sympathetic nervous system, turn that fight or flight response on. Are you able to activate your entire body? Or if we're telling you to activate your sympathetic nervous system from your brain, are you just in control of everything above your level of injury? What happens when we activate your sympathetic nervous system below the level of injury? This would be something like dysreflexia. Is it your entire body that you're getting the fight or flight response, or is it just below and above? Again, you've got this parasympathetic output. And finally, what happens when we ask you to turn your sympathetic nervous system off? Can you do that? We've got a whole host of tests that we can do. And I thought this would be a good point to bring one of those up. I think this is an opportunity for all of us to experience some autonomic testing. In the lab, we do this in a very controlled manner, but this is a Valsalva's maneuver. This is a strain maneuver. You take a breath in, and everyone can do this. Take a breath in, and you breathe out for 15 seconds. In the lab, we are measuring the amount of pressure that you're able to generate. You're in a standardized position. You're at a resting blood pressure. This is someone without a spinal cord injury. This is a typical phasic response from a Valsalva's maneuver. Okay, you breathe out, you increase the pressure in your chest, and all of a sudden that squeezes the veins that are supplying your heart. So your blood pressure goes up just a little bit. You got that extra little boost of blood. Now you're continuing to blow out. You've got all this pressure in your chest, and now you're restricting blood from getting into your heart, right? You've got a pressure gradient that you've got to overcome. Your blood pressure falls. Now, in someone without a spinal cord injury, you can compensate for this. You can say, hey, my blood pressure's falling. I don't want that to happen. I'm going to speed my pump up, right? I'm going to make my heart pump faster. And secondarily, I'm also going to squeeze all the blood vessels in my lower extremity. I'm going to squeeze them in my legs. I'm going to squeeze them in my gut. In that case, blood pressure stabilizes, or in this case, goes up a little bit. This is a normal phasic response to an autonomic test. You can imagine that second part, telling your blood vessels that they need to squeeze in your guts and down in your legs, is something that might be really difficult for some people with spinal cord injury. In fact, that's what we see. The first part of the response is still the same. You breathe out, you get that little boost of blood that goes into your heart. Blood pressure goes up. You keep breathing out. Now you're restricting blood from getting in. Blood pressure falls. Your body senses that, tries to compensate, speeds the heart rate up. But if it can't squeeze the blood vessels in the lower extremity, your blood pressure absolutely tanks. And we can quantify that fall in blood pressure. We can quantify how long it takes for your blood pressure to come back to normal. We can quantify your heart rate responses. And from that, generate a lot of data, objective data on how your autonomic nervous system is functioning. Do you have an autonomically complete or incomplete injury? This then kind of gets to the second inflection point. This is not the standard type of data presentation that people are familiar with. These are not tests that people are that familiar with. How do we make that useful? How do we make that useful to clinicians, to patients, to family members that could really benefit from knowing if you've got control of this part of your nervous system? And so this is what we've come up with. This is what we're terming an autonomic phenotype. And this is basically a snapshot of your autonomic nervous system, a little bit different than the Insky picture that we saw before. This is 6 tests of autonomic function. From each one, we get 3 metrics. There are 2 tests looking at above level sympathetic activation. Are you in control of your whole body or just the part above? We do that with putting someone's hand in ice water, assuming that's above level of injury. And by having them do that Valsalva's maneuver that you're all experts in now. We look at below level sympathetic activation by putting people's feet in ice water and filling their bladders up to a constant pressure. And we look at sympathetic inhibition. Can you turn that part of your nervous system off? Looking at things like heart rate variability, blood pressure variability, and giving people boluses of IV medication that increases their blood pressure. That then runs into the problem of, okay, that sounds good, but what's normal? How do I know how abnormal a response is? You just showed these relatively complicated figures of blood pressure tracings. That's going to be hard in clinic for anyone to grasp. So we've performed these same tests on a large cohort of control individuals without spinal cord injury and basically found out what the average response is and how much variability it is. We plot that radially here around the outside at what's called a z-score. So an average score of 0 corresponds to exactly the average response for each one of these metrics. We can then look 2.3 standard deviations inside or outside of that, kind of what that standard range is, and say that should encompass 99% of the variability. More or less, it is a donut. And if you're inside the donut hole or outside the donut, it is an abnormal response. I timed this out with the meal arriving so that everyone gets excited about the donut and the meals arrive. I did not do that. So let's look at an autonomic phenotype for someone without a spinal cord injury. We see this. This is relatively round. This almost kind of looks like a donut. I picked the colors for this one, so you can see the sprinkles on there with the data points. But compare this to an individual with a T8 Asia Impairment Scale A spinal cord injury. So motor sensory complete. Clinically, 3 years ago, I would have said T8 injury should not have any autonomic dysfunction. Remember, anything outside the donut zone is going to be abnormal. And we see that. 14, 15 standard deviations outside the norm. This person is unable to inhibit sympathetic activity. They're unable to activate their sympathetic nervous system below their level of injury at all. When we activate it below, it does not spread, and they don't have kind of a massive vasoconstriction. But this is a snapshot of what this individual's autonomic phenotype is, how autonomically incomplete they are. And as we've repeated this over and over and over again, now in 43 individuals, we're seeing this wide autonomic dysfunction that we're able to capture in just about everyone with a spinal cord injury. There's a few people that have been autonomically incomplete and actually look pretty normal. And that's also a good and important data to get. So what do we do with this? Beyond making pretty pictures and talking about things, I think it's really important that we translate this. And as Matthew alluded to, one hot topic that I think this can apply to is spinal cord stimulation. So Dr. Grahn gave a great talk about all the motor recovery that can happen with spinal cord stimulation. Some very smart people about 5 years ago thought, well, autonomic function is really important. Can we actually look at if spinal cord stimulation is helping that as well? And in study after study after study, they've shown that, yeah, it can increase blood pressure when you need to. However, all these studies use the same testing paradigm. They have someone lying flat, they tilt them up, their blood pressure starts to fall, they turn on the stimulation, and their blood pressure comes back to normal. The problem with only looking at that below-level sympathetic activation is you don't get that full picture of what's happening. It could be that the signal coming from the brain with the stimulation now is able to be amplified, and all of a sudden you're able to improve your regulation that's happening in your autonomic nervous system. Or it could be that you're causing pain that you're just not feeling and you're getting autonomic dysreflexia. So this is what we did. We went about in 4 individuals testing different types of transcutaneous spinal cord stimulation. We matched individuals with spinal cord injury to uninjured controls. As you can see, this uninjured control at baseline has a nice round autonomic phenotype. We used 3 different stimulation frequencies because different studies use different ones. We wanted to test them head to head. And that normal autonomic phenotype doesn't really get any more normal with stimulation. It's kind of what you would expect. After spinal cord injury, individual has significant autonomic dysfunction at 120 hertz, at 30 hertz, and 30 hertz with a 5 kilohertz carrier frequency. I'd be hard pressed to say that is getting rounder, but that's really, really improving. It definitely helps with that sympathetic activation. So people's Valsalva maneuvers get better when you use the stimulation. But if we look at things like sympathetic inhibition, can you turn that signal off? It actually looks like it gets worse. And what's probably more pertinent is below level activation, putting someone's foot in ice water that doesn't cause dysreflexia at baseline. When you add the stimulation in this small study, caused dysreflexia in those individuals. Okay. How does this translate to clinic? Let's go back to these secondary complications. This is something that I'm really excited about. As I mentioned, the motor sensory assessments really aren't great. In clinic, this really forces me to be reactionary. And maybe some of your physicians are reactionary as well. Someone comes in with a urinary tract infection, we say, okay, we can treat that. They come in with 3 urinary tract infections in a year, and we say, that's about average, we'll treat that. They come in with 4, 5, 6. We say, all right, well, this seems to be more of a problem than we anticipate. Let's make sure that you're seeing urology and do some more preventative stuff. That's not catching it until it's already declared itself. That's, 7 months and potentially a bunch of hospitalizations in there. What happens if we're actually moving beyond that motor sensory assessment and using our autonomic assessments? And so this is something that we've recently started doing, looking at those objective autonomic assessments, grouping in a couple surveys of autonomic symptoms, and looking at how that predicts secondary complications. Do autonomic assessments of blood pressure control inform secondary complications, number of UTIs, number of lifetime pressure injuries, how long someone's bowel program is? And the response is actually pretty strong. This is in only 18 individuals thus far, but we're able to predict about 92% of those secondary complications. I have no doubt that this is going to go down as we build a larger cohort. But putting my clinical hat on, I don't really think I need it to be perfectly accurate. What I need to be able to say is, based on your testing, are you someone at low risk for secondary complications? If I can know that up front, maybe we can start to alter care. Maybe you're not someone that has to have a family member get up 3 times in the night to turn you. If any of you went through that, maybe you're not really at risk for secondary complications like pressure injuries. Or are you in the expected bucket? Do we need to approach this, approach your clinical care knowing that you're probably going to have 2 to 3 UTIs a year, but we're not going to get ourselves pinned into a corner where all of a sudden you're having 6 or 7. And very importantly, are you in this high-risk group? Can we identify before all of those secondary complications happen that you're someone that's really set up to it based on the autonomic completeness of your injury? Because if we know that, there's preventative things we can do. We can make sure people are pressure mapped. We can make sure that you're seeing urology. We can give you preventative medications before all those things happen, so you're not having those setbacks. That's the thing I'm most excited about at this point. So I think key take-home points: secondary complications, autonomically rooted complications, really hold people back from reaching potential after spinal cord injury. I think there's more acknowledgement of that, which is really important. Detailed autonomic laboratory testing really holds a lot of promise for key insights into why some of these things happen. And this model of kind of comprehensive autonomic assessments really holds a lot of potential in terms of assessing interventions that might impact the autonomic nervous system and ultimately improve some of these things. So I think we're going to do questions later. My contact information is here at the bottom. Feel free to email me or tweet at me or whatever the equivalent of that is. And thank you for your attention.
You want to cut? It's going to get crowded up here. That's good. Thanks, Ryan. Hey, Lucia or Chuck, can you bring up another lapel? You can use this. You can yell. Oh yeah, really quickly, just lunch is coming out soon. If you have dietary restrictions, you should have a card in the back of your ID badge. Pull that out, put it on your table setting on your forehead, whatever works, to let the banquet folks know what you need. Good to go. Thank you all. I'm anticipating you have some questions. Anybody?
I can't see anything, so you'll have to.
I'll call them out. Do we have microphones out? Okay, John, go ahead, and then we'll go to Ann.
I like it.
Should wear my sunglasses.
So, Dr. Solinsky, great presentation. Really interested in your, the way you're looking at autonomic dysfunctions in a different way. One of the things I was wondering, looking at that model that you've created, obviously looks very useful, but I think you might be missing a couple of spots. I know you're, looking at the hands and the feet and ice to see, I'm guessing if there's a vascular response, but, temperature regulation. You can ask any of the quads in here, they're cold all winter and afraid of dying of heat stroke in the summer. So I wonder if there's a way you could include measures of temperature regulation. And also, we talked about, sitting on toilets for an hour to empty our bowels. If bowel transit time might be another measure you might be looking at in your autonomic measures.
Yeah, those are excellent points. There are definitely more things to be measured that are clinically important. And I think we're still trying to figure out exactly what those things are. In my mind, this assessment takes, not in my mind, this assessment takes about four hours. So we're trying to also be cognizant of, a sweat test, which would be very, very helpful, is quite involved, especially after a spinal cord injury, getting covered in powder and things like that, and then having to wash it off, being put in one of the sweat chambers. It's a process, right? So in my mind, the key is going to be to make a package of tests that is the most informative in terms of secondary complications, that is also the lowest kind of barrier to use. I think the INSCI exam, the Asia exam with the safety pin and cotton swab is a perfect example of something that is very low tech. Its success is probably based on the fact that it can be used, 40,000 times in the last 30 years and generate all that data. Some of these things are not translatable outside of a very, very specific laboratory setting, and figuring out that balance is I think important. I think Dr. Wecht.
Yes, I'm going to weigh in here a bit. Yeah, so it's, obviously very impressive work. We're, the field of autonomic dysfunction after spinal cord injury is just gaining traction. And I'm thrilled to have young investigators like Dr. Solinsky on this. I am the chair or the co-chair now of the International Standards to Document Autonomic Function After Spinal Cord Injury. It's an international consortium of scientists, clinicians who are trying to do or replicate the ease and the practicality of the INSCI exam to determine the degree of autonomic dysfunction in spinal cord injury. Very rudimentary. We're just getting started. The INSCI exam, as many of you know, has been around for 30 years, 40 years. We just started this in 2009. So the question I would have for Dr. Solinsky would be, that's great work. We need this in the scientific community, academic setting, but how do we translate that into clinics that are around the world so that they can perform meaningful tests that don't take time and don't require highly sophisticated technology to get the information, because the autonomic nervous system is very complex. So you could just weigh in there.
Sure, I'll solve that problem. It's a big ask. It's a big ask, I think, of the scientific community. It's a big ask of the clinical community to buy into some of these measures. And I think the important thing is laying out what the minimal necessary tests would be, how long they take, and what the payoff is, how this is going to improve the patient interaction that I'm having in clinic. How is this informing care? It's unclear what those tests are. We've piloted a few of them, thinking maybe we can do ischemic hand grip, which is blood pressure cuffs and a blood pressure monitor, and block off blood flow at people's arms. And it was a huge failure. I'm not sure what it is, but it definitely, there are minds that are focused on figuring out how to bring this to clinic as we become more and more informed of just how important these measures are. Sorry, that isn't actually fixed.
No, but it's.
Anne?
Yes, hi, I'm Anne Vanderwinkel. I'm assistant professor in the Division of Physical Therapy here in the Department of Rehab Med at the University of Minnesota. And I'm a 48 years old, 5.54. And so I've been doing research for 25 years now, and the last 3 years, more specifically, spinal cord injury. And so I've been, I don't have a question, but I do have a suggestion, and this is why I like this collaborative group. because I think autonomic nervous system is very important, and it's good that people look at outcomes. I've been investigating over 70 people, I think, in the last 2 years with spinal cord injury, and I ask also about an autonomic nervous self-report. And where my research is leading to is more like how to help that, and not just prevent, but just treat and help. And so my research has been in improving body awareness, whether it's with Qigong or whether with a therapy called cognitive multisensory rehab, to improve the awareness of where the body is in space. And what many of those participants report to me is that their temperature has changed, and now they can feel when an UTI is coming up, so they get the clinical care they need. So I think there are ways that are low tech, that don't need any tech, that can help participants early on to improve that awareness so that they don't get to those things, or if they get it, that they feel it quicker and get quicker results. Thank you.
Anita, right here.
Hi, thank you all for from the talks for this session. I have questions for two of you. I'll ask the first one to Peter Grahn. I was curious to know the individuals that you had in your studies. Was it just orthotic effects that they experienced, or was it therapeutic?
So was it, sorry, I missed it. Was it what type of effect?
Was it just orthotic? So the movement that they were getting, was it just while the stimulation was on, or did they actually regain function after a period of time with the stimulation off?
Yeah, so the study that we're conducting right now, they come in, the total number of sessions, we strive for 10 sessions, so 10 days where they come in with stimulation. And afterwards, when we remove the electrodes, of the 14, I believe, subjects we've studied so far, there hasn't really been any observable improvement in their function that they had coming into it. Like, there's been no real change, but I guess it would have been quite surprising if they would have, after only 10 sessions, just based on the literature and what we've seen in our pilot study of 2. Takes time.
Okay, thank you. And then my next question is for Ryan Solinsky. So you mentioned that there was a small number of individuals with spinal cord injury that had ranges within the norm. I'm really curious if you've done any investigations on that group to see why that is. Is there something different with them? Like, can we learn something from that that could be relevant to others that have, autonomic, dysfunction?
Yeah, that's a really good question. Kind of going back on what I said, the lower level injury, individuals lower level injury with more area that they have full control of, have less or have more autonomically incomplete injuries. I guess more characterized as autonomically incomplete. So having a lower level injury seems to be appropriate, and having a motor sensory incomplete also was helpful. That being said, there are a number of individuals that we tested that were motor sensory complete, that clearly had the ability on a dime to squeeze the blood vessels in their legs when they wanted to, that were autonomically complete. And one that stands out is an individual who had been injured about 25 years. He has a T2, Asia Impairment Scale A injury. So he's not moving, he's not feeling anything, but hadn't had a UTI in about 15 years. Never had a problem with his skin, has a bowel program that takes 15 minutes. So, I mean, that data point helps a lot towards the graph that I was showing. But I think it also illustrates that these individuals exist, and really that person is set up for success, much more so than someone with a T2 Asia Impairment Scale A motor sensory complete injury who's in and out of the hospital 5 times a year, who has to be on bed rest, who has a bowel program that keeps them at home for on bowel program day. There's a lot of repercussions to these autonomic injuries that I think we're just now starting to grasp.
Yeah, I'll add to that for a brief moment. I mean, the autonomic nervous system is highly complex. And so the idea that we're going to learn as much as we can from that one data point is probably a difficult ask. But if we could come together as a community and collect more of this data widespread and share it, I think that will really move the needle, and also potentially funding N of 1 studies, understanding, going very deep in a single individual to understand all the nuances of that individual that may or may not be contributing. I think it's something that the funders, I think the community would benefit from, and the funders maybe want to start thinking about the heterogeneity of the condition of spinal cord injury and the rationale behind understanding deeply one person's injury may really benefit the community at large. So just throw that out there.
Sasha.
Hi, Sasha Ripcheski from the University of Kentucky. Very informative talk. Thank you very much, all of you. I want to bring up something that's very common to all the themes here, is that there's lots of logistics about doing the trials and even preclinical studies that are mandated, that you have to go through the rigor. My question, though, comes to the issue of when we hear about all of these talks, it comes to around data points. So I want to ask you all, how much is it difficult to actually collect all the data that you're having, and without just hiding it in your own drawers, share that data and be able to say, hey, don't do that because that group has already done that, or this group has found this, and why don't you find this? And I think, Ryan, your findings are extremely important. I remember 20 years ago when I gave a talk about autonomic dysreflexia, besides the fact that I experienced it myself daily, I was the only one in the audience. There are very few. To hear this now and know and recognize is amazing. But what I would caution you is that as you go forth with your interpretations, please share. Because I understand pre-publications are important, but it's also important for the field to get a sense of where you're going so that we can address these same issues, because I don't want things to be redone that have already been done. So that's not really a question. I'm just putting that out there for you all.
I'm sure I speak for all of us. We could not agree more. This has become a huge current topic in the field that has reached a tipping point. So more data is being shared. The foundations of this problem are partly the incentives do not align with society's incentives. The incentives of academics do not align with this society's incentives. So, but it's changing. It's changing. And I have to say, funders are trying to change it. NIH is mandating this. I have to say, I'm academic, but our institutions are changing the slowest. The funders are changing in response to this need to share the data, because it's the only way for us to learn from the data we're generating and not publishing.
It's built for another purpose, and it's hard to rebuild and renovate for a different purpose.
But I think packaging the data and in a way that's organized and accessible to others is not a trivial task either.
Yeah.
Like, I mean, if you're going to mandate that all your data be shared, like who on the team is going to do it if you're spending all day collecting that data? You know, there's got to be some extra support to do it.
Well, and one of the complicating factors, like I know, I don't want to talk too much, but you know, there was an effort on our part to help facilitate neuromodulation scientists around some of these issues, like what are you measuring? How are you measuring? How are you profiling? And later today, we'll hear from Russell on the Open Data Commons. And there's a very pointed effort right now with one lab doing neuromodulation studies to start crafting that Open Data Commons for spinal cord stimulation. Because Jill, what you said about heterogeneity is really problematic for our community. There is so much, you all know, look around in the room. What variety we have, from high-level injuries to low-level injuries to incomplete to complete, like nature of the injury, like it's crazy. And so it's really complicated to deal with when you're asking these kinds of questions. Zach, I know you have a question.
Hello, everyone. Zach Wagner from Iowa, 24 years old. I have a C4 AGB complete injury. My question is for Mr. Goldsmith, I think his name was, on TRT or testosterone replacement therapy. Just kind of a two-part question. I was just wondering if he has any data if anybody young were to take this, if it was to affect sperm count or possible longevity of it in the future if I wanted to have kids, as well as the next part of if you had a certain time to start it, whether you have a high sperm or high testosterone level, medium, or if there's a certain age that you would recommend for anyone taking that. Thank you.
Thank you so much for the question. So I think an important thing is to get the levels checked. And something that's commonly done is just checking total testosterone, but there's a whole profile that needs to be looked at. So looking at sex hormone binding globulin to determine free testosterone, albumin bound testosterone. This is really important just to find out where your levels are or your concentrations are. But then there is, with most delivery formulations of testosterone replacement therapy, you are providing chronic negative feedback and you're shutting down your gonadotropins, and therefore you could be infertile from that. That's why I'm pretty excited about this intranasal formulation, because it's delivered 3 times a day, 2 to 3 times a day. It doesn't chronically provide negative feedback to the system, and limited evidence seems to suggest that it could maintain gonadotropin levels and maintain measures of fertility. And so I'm pretty interested to go down this road and see the effects of intranasal testosterone. But yeah, I think it's important for everyone to get their levels checked, because it can affect so many different bodily systems.
Okay, one more question from the live stream. Kristen.
From Krista Adamson on the live stream, Dr. Solinsky, have you noticed maybe even a gut reaction to age-related AD differences pre-post-menopausal, male versus female, or years post-injury changes, like less sweating?
Yeah, that's a great question. I was actually talking to someone earlier about this today. Aging with a spinal cord injury is a whole new experience that people get to go through. There's definite changes that people have, not only in terms of motor function that can change, not only in terms of spasticity, but also, yes, in autonomic function. People develop new symptoms, things like sweating, especially usually require some sort of workup just to make sure that there isn't syrinx or something that's forming inside the cord, which is something that can also happen more chronically. But yes, there is age-related changes that happen pre-post menopause. 20, 30 years out, all of a sudden people have things that they have to relearn in their body.
Okay, thank you all.
Good to see you.
Okay, now we're just gonna have lunch, right? Thank you all. Enjoy your lunch. Talk amongst yourselves. And we will be back to take this. So keep some of these questions in mind, because the next set of presentations, you'll hear from several therapists working in the clinic with people who have stimulators implanted. And then we will have a panel of people with spinal cord injuries who have experience with transcutaneous and epidural stimulation, talking about their experiences. So think about everything that we just talked about in the last hour as you will listen to them after lunch. Okay, enjoy.